Welcome back, guys. In this video, we're going to be going into more detail on glycolysis. So if you haven't seen it, check out the playlist. There's a video right before this where I do an introduction to the entire concept of cellular respiration, and we talk about redox reactions. So watch that first. It'll make more sense going into glycolysis. So big picture, I think with all this, it helps to have context. What we're doing in glycolysis really just means we're taking in glucose as an energy source. And glucose is a 6-carbon sugar, right? Those carbons, remember, we don't want the carbon, we just want the energy. And so your goal in all of this is to take that glucose and go through a series of steps that are oxidizing glucose and all the molecules that it breaks down into. So these are all oxidation steps, right, where we're trying to strip energy off of glucose as much as possible. In the end, we're going to end up with a few different things. You're going to make a couple of ATP molecules directly, right? This is called substrate level phosphorylation because we're just transferring some phosphates from these substrate molecules to ADP to make a little bit of ATP. It's a pretty small amount. Your net gain at the end of this is actually only 2 ATP because even though we make 4, right, 2 plus 2 here gives 4 ATP. We had to spend 2 ATP up here, right? And so the net gain is 2. Invested 2 at the beginning, get 4 back at the end. You wind up 2 up in total. All right, so we make a little bit of ATP. We also make a few of these NADH molecules. And remember, NADH doesn't give you any payoff immediately, but eventually, as long as you're aerobic, that NADH will make its way to the electron transport chain because that is what's taking the electrons, right? To convert NAD plus into NADH, we had to add a couple electrons. Those electrons are what are being handed off over here to start the electron transport chain, right? So we make a little ATP directly. We make some NADH that we'll use later in the electron transport chain. And at the end, we wind up with 2 of these pyruvate molecules. Remember, glucose was a 6-carbon sugar. Pyruvate is a 3-carbon sugar, so it makes sense that we have 2 of them, and that makes sense with the name. Glycolysis, right? Glycolysis. Glyco always means carbohydrates, right? Like glycogen. And lysis means to break apart or break in half. And so we are very literally taking one 6-carbon glucose, breaking it in half into 2 3-carbon pyruvates, and getting a little bit of energy out of it along the way. Now, this pyruvate still has a lot of energy left in it, and so assuming we're aerobic, at the end of glycolysis, you're going to shuttle that into your mitochondria, turn it into acetyl-CoA, and have that enter Krebs cycle, or you might hear it called citric acid cycle. And so pyruvate isn't the end of the line. There's still a lot more energy to be had, so think of glycolysis as step 1 in this entire process. Right. And this is showing the same thing in a slightly more simplified schematic overview. This is our 6-carbon glucose. Remember, when you see these diagrams, if you haven't had a chemistry class yet, in these hexagon or pentagon-shaped sugars, anytime you see a ring, each corner of the ring represents a carbon. So we have 1, 2, 3, 4, 5, and then this is 6 carbons in glucose. And this is giving you your summary. These ATP that are being put in are sometimes called the investment phase. We're investing 2 ATP to get the reaction started. And then later on, once we've split in half, right, so this is our 6-carbon glucose. When you see this fork in the road, that means that we're now branched into 2 3-carbon molecules. This is the energy extraction phase, right? So we invested a little bit at the beginning. This is the extraction, or you might hear some people call it the payoff phase, right? This is getting your energy back out. We get 2 NADH, and we get 4 ATP, right? That gives us our net reaction, right? Put 2 ATP in, get 4 back, so we've got a net of 2 gained there, and we have a gain of 2 NADH. And then in the end, you have these 2 identical pyruvate molecules, and we can do 2 different things. If you have oxygen, right, those pyruvates are going to go into Krebs cycle in the mitochondria. If you don't have oxygen, they're going to go to fermentation to make either lactate or ethanol, depending on the organism you're in. Those are the possible fates. If we have oxygen, almost all organisms prefer to go the Krebs cycle route because you get a lot more ATP that way. But some, like bacteria that don't normally have oxygen in their environment, they'll do the fermentation thing. Or like humans, if you're exercising and you're working out really hard and just don't have enough oxygen, your muscles can also do the fermentation thing. All right, so let's zoom in a little bit closer. This is where different classes will involve different levels of detail. Right? How much do you need to know about these individual reactions? That depends on your teacher, professor, what they want you to know. But I'm going to walk through some of these steps and at least give an overview. Okay, remember, when you see these diagrams, all it's telling you is each step in the process is one reaction, and every reaction requires an enzyme. Right? So in step 1, this hexokinase, this is the enzyme. And the reaction that's happening is we are attaching a phosphate to glucose. So for the future, whenever you see the kinase ending on an enzyme name, kinases are enzymes that add phosphates. So what is hexokinase doing? Well, it's taking glucose as its substrate. Right? Makes sense. Hex means 6. Glucose is a 6-carbon sugar. And we're transferring one of the phosphates from ATP onto glucose at carbon number 6, right? That's why this product is glucose 6-phosphate, right? The names seem kind of crazy, but they really do make sense if you look closely, right? From there, enzyme number 2, phosphoglucose isomerase. An isomerase is an enzyme that converts a molecule into an isomer, which just means the chemical formula doesn't change, but we're rearranging the bonds. And so glucose and fructose are isomers of each other. The phosphate is still there. But if you look closely up here, we've rearranged the positions of these bonds between carbon and oxygen. That's why it's an isomerase. Okay? From there, phosphofructokinase, same deal. Phosphofructokinase. It's transferring a phosphate group to fructose, making the enzyme a kinase. And so while this molecule is fructose 6-phosphate, this one, fructose 1,6-bisphosphate, is telling you that now you have 2 phosphate groups. All right, so in the process, we've spent 2 ATP. This one back here is number 1, this guy is number 2, and those 2 phosphate groups that we pulled off of ATP are now attached to this fructose 1,6-bisphosphate molecule. So in all of this, we're just transferring chemical functional groups around to these molecules. All right, a couple things to add. This hexokinase molecule, a lot of times this is called the committed step of glycolysis because once that phosphate group is added, glucose 6-phosphate can't leave the cell. It's basically trapped inside the cell, can't get back out, and so you're committing to either using it for glycolysis or it could be stored as glycogen at this point. Okay, so once we're at step 4, this fructose bisphosphate aldolase is an enzyme that actually does the splitting, right? So now at this point, we're taking this 6-carbon molecule, we're splitting it in half into 2 separate 3-carbon molecules. And they wind up as 2 slightly different products, right? This dihydroxyacetone phosphate looks slightly different than glyceraldehyde 3-phosphate, but there's an enzyme that can convert one to the other. And so step 5 is just converting this molecule into G3P. This glyceraldehyde 3-phosphate you'll see written as G3P a lot of the times. And that glyceraldehyde 3-phosphate is what enters the second half of glycolysis. Okay, so just to be clear, they're showing, to make the drawing simpler, we're showing one G3P molecule entering the second half, but the 2x is telling you that for every 1 glucose you started with, there would be 2 of these molecules, right? Because we split it from 6-carbon into 2 3-carbon pairs. And now we're entering the payoff phase, right? Up here, we invested a couple of ATP to get the reaction going. Now we're going to get it back out. So thing 1 is you're going to break off this phosphate, and in the process, we're going to transfer an electron to NAD plus to make NADH, right? So in this case, we would say the G3P is being oxidized because we're removing its electron. The NAD plus is being reduced because it's taking the electron, and our product, NADH, right? That NADH, remember, eventually, if we have oxygen, can go to the electron transport chain to make more ATP. So you're not getting ATP out of it immediately, but eventually you will, right? Also pay attention to the fact that in glycolysis we produce protons. Those protons will also be used in the electron transport chain if we have oxygen later. If you don't have oxygen, those protons are what can make you acidic. That's the burning acid feeling that you get during a really hard workout when your muscles start to have their pH drop lower and become acidic. Okay, the rest of these steps, same concept. I'm not going to go into every little detail, but we transfer phosphates to ADP to make some ATP at 2 different steps. These steps are both called substrate level phosphorylation, and what that means is substrate level because the phosphate begins on the substrate. This 1,3-bisphosphoglycerate, that is the substrate molecule, and we're transferring the phosphate directly from the substrate to ADP. This is different than oxidative phosphorylation, which is going to come later in the electron transport chain in mitochondria. So when you see that term, substrate level phosphorylation, that's glycolysis, direct handoff of a phosphate from the substrate to ADP to make ATP. Okay? And at the end of all of this, we've got a handful of things now. Okay? We have the NADH that we made up here. We have the ATP that we made over here. And of course, we have pyruvates. Remember, you've got 2 of them, right? 2 3-carbon molecules of pyruvate because we started with 1 6-carbon glucose and broke it in half. They're just only showing you one of the glyceraldehyde 3-phosphate G3Ps entering over here. Okay, so once we have this pyruvate, our next question that we have to ask is, do we have enough oxygen? Because remember, if you have enough oxygen, you're going to wind up going into mitochondria and using that oxygen for a Krebs cycle, right, an electron transport chain. So in this graphic, we're assuming that you have O2, right? Oxygen is needed right here in the electron transport chain for the very, very final reaction of this process. If you don't have enough oxygen, that pyruvate is going to have to be routed somewhere else. Okay? And so when you hear the terms aerobic and anaerobic respiration, aerobic just means you have oxygen in your mitochondria, and you'll send as much of that pyruvate this way as you can. If you don't have oxygen, or you don't have enough of it, you're going to have to go through fermentation. Okay? And so these 2 terms, anaerobic respiration and fermentation, they go together, but they're not exactly the same thing. Right? So glycolysis all by itself, that is anaerobic respiration because we don't directly need oxygen for glycolysis. Fermentation is what happens to that pyruvate if you don't have oxygen available. Right? So we have 2 major options to work with here, and different organisms do different things. So in either way, we have pyruvate that came off the end of glycolysis, right? We don't have oxygen, so it can't go this way. And you can convert it into one of 2 different things. Some organisms, like humans, convert pyruvate into lactate or lactic acid. And others, like some yeast and some bacteria, convert pyruvate into ethanol. Right? Ethanol is just drinking alcohol. It's the reason that fermentation of sugar from grapes or starches from wheat or barley can make alcoholic drinks. So in this process, there's one other important thing that happens. In order to convert pyruvate into either lactate or ethanol, you need to use an NADH, and that NADH gets converted back into NAD+. That sounds like it's going to be a bad thing, but it's actually a good thing, because remember, you need NAD plus as a reactant for glycolysis. And so the reason that we convert pyruvate into lactate or ethanol, one is you have to get rid of the pyruvate. You can't just let it build up forever. And thing two is we say that we regenerate NAD plus, because if you don't have NAD plus, glycolysis won't function, right? And remember, you don't really have any use for that NADH anyway, because normally you would use it to take electrons to the electron transport chain. But if you're anaerobic and don't have oxygen, the electron transport chain can't run anyway, and so it doesn't really matter. Okay, so when an organism or a muscle is anaerobic, that just means we're churning through glycolysis, we're using a bunch of glucose, you're getting a little bit of ATP out of it, it's better than nothing. And then as a way to get rid of that excess pyruvate that you end with, right, you ferment it into either lactate or ethanol, using NADH as an energy source and regenerating NAD plus to make sure that you have more available to keep glucose churning through glycolysis. All right. Okay, hope this was helpful. The next step in this process is going into mitochondria to convert our pyruvate into acetyl-CoA and enter Krebs or citric acid cycle. So follow along in the playlist. That's the next stage in all of this. And subscribe to the channel to make sure you get all these. The videos work so much better when you watch them in sequence.